Welcome to a new video on microscopy. The invention of the microscope opened up the microscopic world. It caused a sort of revolution in scientific world. Zacharias Janssen made one of the earliest compound microscopes in 1600.
Welcome back and welcome to a new video on microscopy. Microscope is a very important instrument in biology, with the help of which we can see the objects that are normally not visible to naked eye. The resolution power of a normal eye, human eye, is 0.1 millimeter. So we need to use a microscope to see anything beyond that. Basically, there are 5 types of microscope. In fact, there are many other types also. The simple microscope, compound microscope, electron microscope, stereo microscope, scanning probe microscope. These are the 5 basic types of microscope. In this class, let us only see the details about the compound microscope and electron microscope. We will also learn the differences between simple compound microscope, which is also called the light microscope. The other one is called electron microscope. In the light microscope, you use the light as the source of energy. In case of electron microscope, we use electrons as the source of energy for seeing the objects. Compound microscope has 2 sets of lenses. These are called ocular lenses. In fact, this is a binocular microscope. In a normal microscope, we have only 1 set of ocular lenses. In addition to that, we have the objective lenses. See, the object is kept here, the slide is kept here, in which you may have bacteria or blue-green algae or any other material that you want to see it in a microscope, and the object is observed through 2 sets of lenses. This is the first set of lenses called objective lenses, where we have different options, the normal one, and we have the oil immersion, low magnification, high magnification, and here we have ocular lenses. So your section is ready. Now you can observe it under microscope. This is the stage of the microscope on which you can keep the slide. Alright, I have kept it. Next, you select the low power, move it up and down. You can move it up and down by using this knob. Okay, and you select the low power. There are different objective lenses, so you can use the smaller one, okay, which is the low power, and then you can move it up and down by using these knobs. Minor adjustment, major adjustment. This is the bigger one is major adjustment, this is minor adjustment. Now what you require is, you require about 1 centimeter gap between the slide and the objective. And then you see it here, see it through the ocular lens, and use this knob, okay, and move the slide up and down, front and back, so that it comes under focus. Yeah, the slide is focused. So through ocular lenses you are seeing the object, and the image of the object is passing through the objective lenses. And below the stage, this is the stage, below the stage there is a diaphragm and a condenser which will actually control the intensity of light, and at the bottom we have illuminator, the light source. This is a light microscope because you are using light as the source of energy. This is the arm with the help of which you are carrying the microscope from one place to the other place, and there are 2 adjustments. This is the fine adjustment, this is the coarse adjustment. Initially you adjust the object with the help of coarse adjustment. Later, if you want fine tuning, you can use the fine adjustment. This is the base of the microscope, and this is one of the first microscopes that was invented by Antony von Leeuwenhoek in 1600, and he is considered to be the father of microbiology because he is the one who first popularized the microscope. A light microscope is an optical instrument used to view the objects too small with the naked eye. The maximum magnification of an optical microscope is typically 1000 times. and the resolving power of a microscope is about 0.25 micron. That is also 0.002 millimeter. At 400 magnification, you will be able to see 0.45 millimeter or 450 microns. At 1000 times magnification, you will be able to see about 180 microns, the objects of 180 microns. And if you want to see the simple calculation, you are actually using two sets of lenses, which I have just now showed you in the previous diagram picture. There are objective lenses and ocular lenses. The lenses that are used in the objective lens and ocular lenses have to be multiplied. For example, if you are using 10x of objective lens and 10 of ocular lens, the magnification is 100 times. Similarly, if you are using 100 objective lens and 10 ocular lens, the magnification will be 1000 times. This is the electron microscope. And this is the difference between light microscope and electron microscope. In light microscope, you are using light as the source of energy, which is passing through the objective lens, and later it is absorbed under the ocular lens. There is a condenser also. This is the light source, and there is a condenser. This is the specimen through which the light is passing, and it is passing to the objective lens, and it is absorbed under ocular lens. So this is the outline of light microscope. Whereas in case of an electron microscope, the source is electrons, high energy electrons, and there are condensers which will condense the electrons and pass it through the object or the material, and the object is not directly seen as you see in case of light microscope. Light microscope, illumination source is light. In case of electron microscope, the illumination source is electrons, and specimen. These are the differences between a light microscope and an electron microscope. In case of light microscope, the source of energy is light, and it is electrons in electron microscope. Specimen preparation takes usually a few minutes in case of light microscope, whereas in case of an electron microscope, the specimen has to be prepared. It will take a lot of time for the preparation of the specimen. Only dead or dried specimens are used in case of electron microscope, whereas in case of a light microscope, you can see the living material. Condenser in case of a light microscope is there, which will condense the amount of light, and objective and eyepiece lenses are made up of glasses. Whereas in case of electron microscopes, all lenses are electromagnetic. And in case of light microscope, specimen is stained by using some colored dyes. Whereas in case of an electron microscope, specimen is coated with heavy metals in order to reflect the electrons. And as I mentioned already, the resolution power of a light microscope is about 0.25 micron, whereas in case of electron microscope, the resolution power is 0.001 micron. That means light microscope can maximum magnify up to 1000 times, maybe up to 1500 times, whereas in case of electron microscope, the magnification is 1 lakh. Nowadays we have electron microscope which can magnify up to 5 lakh times. So the magnification in case of light microscope is maximum 1500 times, and magnification in case of electron microscope is about 5 lakh times. Vacuum is not required in case of a light microscope, whereas vacuum is essential in case of an electron microscope. The image in case of light microscope is directly seen through your eyes, through the ocular lens, whereas in case of electron microscope, the image is not directly seen. It is seen on a fluorescent screen, or it may be a photographic film. The image is taken in a film and that is observed later. So these are the differences between light microscope and electron microscope. Basically, there are three types of electron microscope: scanning electron microscope, transmission electron microscope. The third one is a recent one called cryo-electron microscope. Let us only look into scanning electron microscope and transmission electron microscope. In case of scanning electron microscope, the electrons are made to fall on the object, and they are allowed to scatter, and the scattered electrons are detected later, and the image is taken from the electron that are scattered from the object. The image is either formed on a TV screen, on a photographic plate, or anything else. Whereas in case of a transmission electron microscope, the electrons actually pass through the object. The electrons are detected and image is formed from that. So in case of scanning electron microscope, electrons will fall on the object and they scatter. Once the electrons scatter from the object, the scattered electrons are observed to make the image or to obtain the image. In case of transmission electron microscope, the electrons are made to pass through the object, and after the electrons passing through the object, the electrons are used to make the image or form the image or create the image. And in case of scanning electron microscope, the object need not be cut into thin sections. You can see the small objects like pollen grains, spores directly. The surface is observed. The structure of the surface of the molecule or any object is observed. Whereas in case of a transmission electron microscope, the object has to be cut thin. Very thin section is required for observing under a transmission electron microscope. The surface is actually polished with powders in case of a scanning electron microscope, whereas in case of a transmission electrons, tiny particles are observed, grain boundaries are observed, the objects which are very thin, and the electrons pass through them, and the internal structure of those objects is very clearly seen. So basically, in case of scanning electron microscope, the surface of the object is seen. In case of transmission electron, the internal structure of the object is seen. And the resolution power of a scanning electron microscope is lesser as compared to transmission electron microscope. These are the two basic types of electron microscope. As I mentioned, there is one more, the third one, called cryo-electron microscope. Thank you very much for watching this video. Please open the description box and you will find the details about this video. We'll meet again in the next video with some other topic. Thank you very much.